And then we will talk about experiment 17. All right. So experiment 18, a couple things. First of all, you should get your GCMS data from your lab prof, okay? So get that this week if you haven't already. In lab this week, you're going to process your NMR data, so you'll leave lab this week with your NMR data, okay? One of the things you need to do is compare your IR, NMR, and mass spec data to known spectra once you figure out what you have as an unknown. Page 1616 is a good page for that. But keep in mind, we were not using a melting point or a boiling point to figure out what these unknowns are. So you shouldn't be using a melting point or a boiling point. The intent of these unknowns is you're using all the data that you have collected to figure out structurally what that unknown is, okay? So you need to use all that spectroscopic data, your IR, your NMR, your GC mass spec, as well as the data you collected in lab, your solubility tests, your chemical tests, to put together to figure out structurally what that unknown is, okay? So this shouldn't be a hunt and match up spectra exercise. It should be use the data, figure out what you have, use the known literature spectra to confirm that, okay? So you don't want to go looking for it in the literature first. You want to figure out what you have first, all right? And if you do it backwards, it'll be very evident in what you write up for your conclusions. So make sure you go about it in the proper direction. But otherwise, just make sure you get all of your data that you need for experiment 18, because that all will be due next week. So you'll have to put all that data together. And it's going to be more complex than experiment 6 was, because there was more data collected, okay? All right. Let's look at experiment 17. So experiment 17 is going to use a lot of things that you have not encountered yet, including the reaction, the Wittig reaction you have not covered yet in lecture, but you will soon. Have you guys finished with NMR yet? You're still doing it? Still on NMR, so as you finish with NMR, then you will quickly get to talking about carbonyl chemistry, and that's where you're going to talk about the Wittig reaction. A key piece to the Wittig reaction is what is called the ylid, okay? And what this ylid is it's a species that exists where we've got two charges present on adjacent atoms, okay? One being a phosphorus, one being a carbon. We've got positive on the phosphorus, a negative on the carbon, all right? And so this exists as this entity with adjacent charges on adjacent atoms, and this is the key to making this reaction work. All right. So this ylid is formed by taking a phosphonium salt. Okay. So taking a salt with triphenylphosphine oxide or triphenylphosphine in it. Okay. And you react it with a base, and then you will form this phosphorus ylid. All right, and so that's what we'll work through those steps here in a second. But that's what you're going to do in the first step of your reaction, is formation of this ylid. All right. Then what can happen is that ylid can go on and react with an aldehyde or a ketone. So that negative carbon can then go on and attack the carbon of a carbonyl, because the distribution here electronically is you've got your electronegative oxygen, your less electronegative carbon. Okay, so it's going to be like the Grignard reaction, where that carbon of the carbonyl is susceptible to a nucleophile attacking it. Okay, So our nucleophile is going to be from our ylid, that carbon will attack our carbonyl, and eventually what we will form is an alkene, and then a byproduct of this reaction is triphenylphosphine oxide. Okay? So this reaction is really useful because it's another carbon-carbon forming reaction. There are multiple ways that you can get to the ylid, which then can react with the aldehyde and the ketone. Kind of a downfall of this reaction is this triphenylphosphine oxide, because you're going to make that byproduct and you then have to get rid of it, okay? But this is a reaction that's used research-wise, it's used in industry, all sorts of different places because of the utility of it, especially with the phosphorus ylid. So I'm going to talk about this part here in a little bit, but let me talk about forming that ylid first. So what happens when you form the ylid is you're going to utilize the electronics of what you've got present to either make this stable or not, all right? So we've got this triphenyl phosphonium salt, okay? And this can be formed with taking a compound and reacting it with triphenylphosphine, okay? And then you get this salt. So that makes this part pretty easy. If it can react with the triphenylphosphine to form the salt, you can make the salt, all right? And then you're taking it on with the base to then make the ylid. Couple keys to this, and so the reason I'm using the specific letters here is I'm going to explain some of the things that are key to this. If what you've got for your Y and Z groups, okay, so when you react it with the base, we're going to take this proton, all right? That's how we get the charge here. Your Y and Z groups, if they're either protons or alkyl groups, this is not going to be a very stable situation, okay? So your ylid won't stick around very well, and it's considered to be unstabilized if Z and Y are alkyl groups. And so what you have to do in that case is you would have to make the ylid and use it right away because it's not going to hang out for very long. But if you make these groups something else, especially if you get these two groups to be electron withdrawing, then you can get what is called a stabilized ylid. And so that you can even, Aldrich will sell some stabilized ylids. Like some of these are so stable, you can put it in a jar, put it on the shelf, and it'll stick around, okay? We are using, what we are going to form has an electron withdrawing group on it, but for what we are doing with our reaction, we're still forming it and then going on and immediately using it in the reaction. But because we have that electron withdrawing group on it, it's going to be more stable than something that does not. And usually when we're talking about things being more stable, what does that usually come from? Resonance. And so that's where this comes from, okay? Is unstabilized ylid, you don't have as many resonance structures versus a stabilized ylid. The resonance structures that you can form, there's greater resonance structures that provides more stability, okay? And so that is the case for what we are going to use. In your lab manual, it shows you in figure 17.3 this comparison. This is the ylid we are going to use. So it shows you that there's more resonance structures present, and therefore it's more stabilized, all right? So I'm going to get back to this here in a second. But what happens, besides the illid, another kind of vocabulary word for this reaction, is in the process of the illid reacting with the aldehyde or ketone, what is formed, so as we have the nucleophilic attack of our carbonyl, we form this intermediate, and then it can go on. So we've got this intermolecular oxygen-phosphorus bond that can be formed. And what is formed is a beta-ene, okay? And so this structure here is a beta-ene. And you'll see this again when you guys cover it in lecture. But we get this beta-ene that then, so four-membered ring, typically not going to stick around for very long, right? So we get this intermediate. It then decomposes, and what happens when it decomposes is the oxygen goes with the triphenyl group. We get our triphenyl phosphine oxide, and then we get the double bond formed for our alkyl group, okay? And so then our Y and Z came from our illid, our R and R prime came from our aldehyde or ketone, okay? And so then we join those pieces together, all right? And like I said, you'll go into more detail with this when you cover it in lecture, but this is kind of the basics of the reaction, so you have an idea of what's going on, all right? And it is, if you want to look in McMurray for more information, there's page 720 of the 7th edition talks about it. So for our particular reaction, we've got this triphenylphosphonium salt, which has, if you look in your lab manual, it has like a 26, I don't know, it's a long name, okay? So we'll know it as our triphenylphosphonium salt, okay? And it's a bromide salt. We are going to react it with sodium methoxide in methanol. So our base is our sodium methoxide, all right? So the sodium methoxide is going to deprotonate our salt, and then we form our illid. So we can draw it as this resonance structure, but we can also show it, here is taking this resonance structure and showing you where that negative and positive is. So here's our negative, here's our positive. This is the carbon that's going to attack our the carbonyl of our aldehyde, okay? So we form this, so that's why in your procedure it talks about taking the salt, you dissolve it in methanol, you add the sodium methoxide, you stir it for 5 minutes. That stirring is forming the illid, okay? So that you can get it formed. So then when you have the aldehyde added, so hydrocinnamaldehyde, so what do you think it's going to smell like? Cinnamon. So it's very, very close relative to what, you know, the scent and what you add in for cinnamon tasting. So it smells like cinnamon, so it doesn't smell too bad. We will add this to your reaction, so we're going to have to add 0.7 mils. So we'll, your TA or prof will add it with the syringe, okay? And then it will go on and do the reaction to form your product, okay? This is fairly quick. This we are going to let reflux for an hour. During this reflux period, we're then going to teach you how to process your NMR data, and so that's where you're going to learn how to do that. We'll utilize that hour and teach you how to process your data, so that when you leave this week, you'll have your NMR data for experiment 7, experiment 18. Make sure you pay attention to that though, because this is our one time of showing you how to process the data, and then you're going to be on your own. We're going to turn you loose to do it on your own from here on out, all right? What we will form then is our cis and trans. We're going to have this mixture of cis and trans product, and then the triphenylphosphine oxide. The workup that you're going to do at the end, we already have our product in there. The workup you're going to do is trying to get rid of that triphenylphosphine oxide, okay? And so you're first going to do a distillation, simple distillation with methanol. Actually, here, I'm going to back up a little bit. The reaction you're going to do in a hot water bath so you're setting this up for the TAs to watch and make sure everything goes okay for you while you're learning how to process NMR data. So make sure you put enough hot water in there that it's going to last for an hour or more of refluxing, okay? So you're using a hot water bath. Do not use a heating mantle. Use enough hot water and use hot tap water so that it gets refluxing right away, okay? Then from the reaction, you're going to do a simple distillation that you're also going to use hot tap water on. So you don't really want to cool this down because you don't want to lose the heat that you have in there. You just want to very carefully convert your reflux to a simple distillation, all right? All right. Then we tell you want to take off as much methanol as possible. The reason for that, the triphenylphosphine oxide is really soluble in methanol, so you want to get rid of as much as possible. It is hard to get rid of all of it, okay? The other thing that gets a little hard to figure out is your product is a liquid, and so when you're distilling, you're going to get down to where it looks like there's just residue left, which part of it is actually your product, all right? So what we are then going to have you do, and this seems kind of counterintuitive, but it helps to remove the methanol. You're going to take apart your distillation. You're going to add hexane to that mixture that has your product in it, and you may start already seeing solid in it, that's fine. You're going to add hexane to that, and we're going to use what we call a rotary evaporator, which this is a best looking picture, but you guys will see this in lab this week. So this is a really nice tool because it's a way to do a simple distillation under vacuum so it goes faster. Now the reason we are not doing that at this step is we have four rotary evaporators, and methanol takes a little bit to take off, and you already have it good and hot. So it actually is faster for you to do the simple distillation, okay? Then cool everything down, stand in line, wait to be able to use the rotary evaporator, all right? So we're gonna have you do this simple distillation. But the hexane, the first hexane, we're then going to remove with a rotary evaporator. Then you're going to add more hexane. This was to help remove that residual methanol that just is stuck that you can't remove with a simple distillation. All right? So this is to help get rid of the rest of the methanol. Then you're going to have your second hexane portion is going to be used to help filter. Actually, you're going to hopefully first precipitate the solid, which is your triphenylphosphine oxide. All right? And then we will go on and filter that. At this step, sometimes that solid does not precipitate right away. So you don't want to try and filter something that doesn't have a solid, and you don't want to filter an oil, okay? So if after your first hexane that you've then removed with a rotary evaporator, and we want to take down as much liquid as possible without getting rid of your product, you add hexane again and you don't really get a precipitate, see your TA or your lab prof, because we need to make sure we get this precipitate first and then you filter it, all right? So don't filter it until you actually have a solid. If you have an oil, we've got to turn that oil into a solid, all right? So we'll work on it and figure out what we're going to do to make it a solid, all right? So only filter the solid, and you're getting rid of the solid, so what do you use to filter it? Gravity or vacuum? Gravity filtration. Then the solid in that filtrate will go in organic waste. So your triphenylphosphine oxide will go in the organic waste, and the filter paper will go in the filter paper waste, okay, in the reagent hood. The reason for that is it's just really stinky, so we don't want to just throw it in the regular garbage, okay? It's stinky and it's got chemical contamination on it. We're going to put it in the filter paper waste, all right? So then in the end, you will have this hexane solution of your product, okay? Because remember, all along your product is a liquid. You're going to then, again, one more time, use the rotary evaporator to remove the hexane. Now what you probably want to do before that, okay, is you also have to TLC your reaction mixture. You're going to have a solution at this point. Before you use the rotary evaporator, it's probably a good idea to do the TLC first, okay? So I would spot that solution before you remove all of your hexane, all right? And there's two standards for the TLC. So just like we did with experiment 12 last semester, we're going to use standards to help figure out what of your mixture is starting material and what is product, okay? So there's the aldehyde, which is your reactant or product. The hydroxymaldehyde is your reactant, right? And then we also have Wittig product to use for TLC, okay? So you will probably want to, on your TLC plate, have 3 spots, your reactant standard, your product standard, and in the middle will be the reaction or the crude product mixture. Okay. Now, when you are filtering the solid before you go on and remove the hexane, try and do as good a job as possible to get that solid out. Next week, we're going to use what's called column chromatography to purify your product. And if you have a lot of solid in there, it's going to make it harder to do the column chromatography. So you want to try and get as much of that solid out. So first of all, make sure you form a solid before you filter it, and then just make sure you do a good job with the gravity filtration to remove the solid from the liquid before you go on to use the rotary evaporator. All right, and you can, that filter paper, you can rinse with a little bit of hexane too to make sure you get your product out there. All right. So in the end, you'll have a round bottom flask that has a residue in it that doesn't look like very much, but it'll be enough to work with. And then you will have made and developed a TLC plate. I would hang on to that TLC plate because you're going to use it as a reference for your column next week. So I would just, once you develop it and draw it in your notebook and make all the measurements for RF values, then I would put it in a paper towel and save it in your drawer till next week, so you've got it for reference for next week for running the column. Okay? All right. Questions for experiment 17, part 1? Good? Okay. Now what we are going to do is look at a couple examples that will help you with experiment 16. And so I'm going to go through a couple of where you have to calculate chemical shifts, and go through some of those examples. Before that, there's a couple things to note. Number 1A, 1B, and 1C on experiment 16, okay? You need to use altered spectral viewer. And so that is available in the campus computer labs. So even the computer labs we're using this week, 3101 and 33,002, altered spectral viewer is available in those computer labs. So to do 1A, 1B, and 1C, make sure you use that for experiment 16. And it tells you that at the top of the experiment 16 as well, okay? All right, we are going to look at calculating chemical shifts for this compound here. So this is 4-ethoxybenzonitrile, okay? And the first thing we're going to do, look at is the methylene protons, okay? So the first set of protons we're going to look at here is the CH2 of the ethyl group. So to do that, I'm going to turn this down a little bit so you can see this better. To do that, we're using the table for methyl and methylene protons. That's in experiment 16, all right? And so what we need to use is this formula, 0.23, and then we're going to add the chemical shift of X and the chemical shift of Y of the substituents on either side of that methylene, all right? So for the CH2, the two pieces are, we've got basically an O aryl group, and then we also have the CH3. So write out here the start of that equation. We'll start with our 0.23, and then we need to look at what happens with the chemical shift with an O aryl group. And so that is this guy right here, okay? So we add that coefficient to this, to the 0.23, all right? So if it's positive, you add it. So it's positive 2.94, so we're going to add that. The other piece that we've got to look at is what happens when you have a methyl group on it, a CH3 group. And that changes it by 0.68. So we're going to add that to it. And so our calculated chemical shift comes up to be 3.85 for this set of protons right there. All right? Everyone good with using that table? Okay, so now we're going to do it for the CH3 group. So we start with 0.23 again. This time, on one side of the CH3 group is that whole piece. So we've got an alkyl group off the aryl group. And so we're going to add that piece for it. And I'm losing it off I've lost our little piece that I'm looking for. There we go. We don't really have on this table alkyl O aromatic rings. So the best thing you can use is for that CH2, which is not perfect, but the best approximation, is we'll use that we've got an alkyl group there, okay? So we're going to add 0.58. On the other side is just a hydrogen off that methyl group. And so we add 0.34. So the hydrogen in this case does affect the chemical shift, all right? And so we get 1.15 for this set of protons is the calculated chemical shift. Everyone good? Good there? Okay, so now I'm going to look at the aromatic protons. We've got to use a different table to do that, all right? So we need to use the table for aromatic protons. And the way this table works, here's our formula. We're going to use 7.27, and then we're going to add these chemical shifts to it. And we add chemical shifts for what is ortho to the proton we're worried about, what is meta, and what is para. So what you need to keep in mind with this table is this is just the chemical shift for the ortho position, then meta, then para. All right. So for HA, first we'll start with our 7.27, and then we have this nitrile group that is ortho. Okay. And so we've got to add the substituent, or the coefficient for the nitrile group, which is right here, 0.27. The other thing that is ortho to HA is HB. In this case, on the aromatic ring, there is no change to the chemical shift if something is in the ortho, meta, or para position that's a proton. All right? So the protons don't change the chemical shift. But the other piece that we have, besides our nitrile group, is then meta to this HA is we have the ethyl group. And so we've got to use in our meta piece, okay, we got to make sure we grab the right guy here. Our OR alkyl right here at meta is minus 0.09. So that's what we're going to add for the meta substituent. And then we don't, then meta otherwise is itself. It's not going to change its own chemical shift. And para is another HB, so that won't change the chemical shift. So in the end, the calculated chemical shift is 7.45 for these protons. And then finally for HB, we've got 7.27 plus with HB, ortho is the ethoxy group. So we are going to start with minus 0.43. And then with the HBs, meta is the nitrile. So we add 0.11. And again, then para is an HA, so that contributes 0. So we've got 6.95 is a calculated shift for these guys. All right? Everyone good with those? Okay, so let's look at a spectrum just to kind of compare our calculations versus what actually is there, okay? So this is the spectrum for the ethoxybenzonitrile. And so for the aromatic protons, we had HA, we were calculating it to be about 7.45, and it's about 7.548. So it's close. It's not going to be perfectly on the dot usually, but it's close. And for HB, we had 6.95, and it's 6.942. So that one is really pretty close. But even more important, you can see the two signals, and you can determine which signal is which by calculating the chemical shifts, because now you know which one is going to be due to HA from the calculation and which one is due to HB. All right, so that's where it comes into play as far as the aromatics, figuring out which signals belong to which when you've got something like para substitution, where you don't have a splitting pattern or anything like that, where you can figure out what is what, or integration, because they're going to integrate to 2 and 2 and they're going to have the same splitting pattern, all right? For the methylene, which is C here, we calculated it to be about 3.85, and it came out to be about, it's going to be the in between 4.086 and 4.063, so it's about 4.07, so fairly close. And then for D, the methyl group, we calculated 1.15, and it was 1.44, 1.439. So again, fairly close, but not exactly on. But still, you could use it to confirm where those protons were. Another reason why I'm showing you this, besides showing you how the calculations then helped as far as making assignments, is also to show you how to label things on an NMR spectrum, okay? So you want to draw the structure of what you are analyzing on the spectrum, all right? And then label the chemically unique protons with letters, and then you're going to put the letters above the peaks that are representative of those protons, all right? So that is how you will label your spectrum. So it's really important every time you generate, especially for the most part in this semester, we're going to generate proton spectra, not carbon, but you would do a similar thing with carbon spectra, with the carbons versus protons. It's really important that you draw the structure on there and then label the chemically unique protons with letters, and then put the letters above the peaks that correspond to those protons. So you will want to do this with your unknown. Once you generate your own spectrum, you will want to, this will be part of the analysis of your spectrum is doing this, all right? The other piece to it is, remember the table from last week that I showed you for, I showed you what table to use for IR and what table to use for NMR. That NMR table is what you want to use for analyzing your NMR data. The first column of that table is the letter of the proton. We're talking, this is what we're talking about as far as the letter of the proton, all right? Okay, the other piece of what I need to talk to you about, which is in experiment 16, and this is kind of really the only place you're going to get this because they don't really cover it a whole lot in lecture anymore, is how protons on aromatic rings and across double bonds are related to each other and how they affect one another with their coupling in their electronic interaction around the ring or across the double bond, all right? So we looked at, as far as chemical shift, it was not changing for protons to be next to each other, all right? But what happens in the splitting pattern and the coupling that occurs, that does have an effect, okay? And so this is in your lab manual. For the different positions on an aromatic ring, you're going to have the most effect of coupling or splitting in the ortho position. There's usually a 6 to 10 hertz coupling constant involved in that relationship, all right? For meta, it's usually a 1 to 4 hertz, okay? And for para, it's usually 0 to 2 hertz. And you will see some of this in your unknowns. You'll also see some of it in experiment 17's product, and you'll see it in experiment 20. You will see this relationship of the different protons on an aromatic ring, okay? And so the way you can figure out what coupling is present is you actually calculate the coupling constant, okay? And we'll show how to do that next week, all right? So right now, this week, we're going to focus on the splitting patterns, okay? So the other relationship then is, like I said, across a double bond, all right? So we've got our double bond here, and there's different positions across that bond. So we've got vicinal cis, vicinal trans, vicinal, and allylic, which gets a little bit confusing. The vicinal cis and the vicinal trans are closer than just the vicinal, okay? So vicinal cis, vicinal trans, you're just two neighboring carbons, all right? You're right directly across that double bond, all right? And it's just one proton, carbon, carbon, proton is that relationship, all right? Cis are the protons cis to each other across the double bond. Trans are they trans from each other across the double bond, all right? So either they're on the same side or opposite sides. Cis, the relationship, the coupling is about 8 to 12 hertz. Trans, the coupling is 12 to 18 hertz. So again, we'll see this next week when I talk about the product for experiment 17 and what it looks like in the NMR. Then just vicinal, which is not a cis or trans interaction, is then we're not looking across the double bond, but we're looking at the same side of the double bond. Okay, so if we were looking at this proton, the vicinal relationship would then be the carbon of the alkene, another carbon, and then this proton. All right, so it's still 2 carbons away, but it's on the same. It's not across the double bond; it's on the side of it. Do you see the difference there? That relationship is less than these two. It's about 6 to 8 hertz. Okay, and then finally, the last relationship is a allylic. So we're looking at something 3 carbons away. So here's the proton we're interested in. Then we've got across the double bond 1, 2, 3. It's this proton here that can affect the coupling of this proton all the way over here. Okay, and so that's a allylic, either this direction or this direction. That's about 0 to 3 hertz. Right? Okay, so I am going to go through the example that is in your lab manual, how they came up with proposing splitting trees, which is your proposal of what the coupling constants and the splitting are going to look like based on what nearest neighbors are involved in that compound, either aromatic or across a double bond. They use an example where we're looking at what's going on in the double bond, but then we've also got potentially we could also look at coupling here of these 2 protons. But what they are looking at in that example is across this double bond. So it's the trans 2,4,5 trimethoxypropenylbenzene, is this compound here, and we're looking at the different interactions across the double bond. So when HA and HB are trans from each other, they've got a vicinal trans relationship that's 12 to 18 hertz. When they are if they were cis from one another, then you would have the cis relationship. HA has a relationship to this methyl group that's allylic because it's HA, then carbon, 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 and then it's these protons that are involved that will cause some coupling interactions on HA. Versus, so that's a 0 to 3 hertz interaction, versus HB and this methyl group, so it's this same side of the double bond here, but without going across. HB has a vicinal relationship, so that relationship is stronger. It's 6 to 8 hertz. All right. So in the spectrum, what happens is we would say, okay, HA, first of all, had a 12 to 18 hertz splitting because of HB. All right, so we're going to propose that the splitting, the initial splitting, and you start with your strongest coupling constants first and work your way down. So we've got the splitting due to HB that is a trans interaction. It's 12 to 8 hertz. We're split once by that proton. All right, and you're split once because there is 1 proton, but when you split it, you end up with 2. Basically a doublet, okay? So that, have you guys covered the n+1 rule yet? So that makes sense as far as n+1. There was 1 proton, but yet you see 2 peaks there, right? Doublet there. But then this signal goes on and splits more because of that methyl group. And so what happens is it splits once for each of the protons in the methyl group. So 1, 2, 3. So if we take our doublet, split it, we have a quartet. Split it again, then we've got, well, not a quartet. We've got split it as a doublet, then we've got a doublet of doublets, right? Then we split it again, we've got a doublet of triplets, and then we split it again, and we've got a doublet of quartets. So that's where those 3 protons from the CH3 group come into play. 1, 2, 3. So in the end, what you're proposing is you're going to have a doublet of quartets due to the proton that was trans from HA and then the relationship of the methyl group. This is really big relative to what's happening from the H's of, or the protons of the methyl group. Do you then see that? Or we have that big coupling from trans, and then we go down to really small because it's a allylic relationship of HA to that methyl group. Good with that? Okay. So then for HB, it gets a little more complicated. We've got a trans relationship to HA. Okay, so we've split into our doublet from HA. Still about 12 to 18 hertz. But that methyl group and HB have a stronger relationship. The coupling constant is greater because it's a vicinal relationship, not allylic. So it's 6 to 8 hertz. And what happens is we still get split once by the methyl group, twice, 3 times. There's 3 protons, so we're split 3 times. 1, 2, 3. We end up with a doublet of quartets. But what happens is because of this coupling being so strong, instead of you having doublet of quartets and they're hanging out separate from each other and you can find them really easily, you end up getting this crossover of the signals, okay, because the coupling constant is so strong. So then what you would have to do here is show that you've got this where the two sides of the signal cross over each other, all right? And that's from that coupling constant being so large. So then the other part that they show you in the lab manual is then the actual proton NMR. You can see that interaction. That is what you are seeing in the peaks that are shown. And so this is how you would draw the splitting trees above those signals. And that's what you will eventually need to be able to do with experiment 17, is draw your prediction of the splitting and teasing out the splitting from what you see in the actual spectrum. Okay? So we'll go through this again more next week, but this is where this comes from. We're going to go through it in specific detail for experiment 17. Right? But this is what you're looking at when you're predicting coupling interactions and splitting interactions, and the splitting of